All right, it is my pleasure to introduce Dr. Brittany Howell, Assistant Professor in Neuroscience and Human Development from FBRI and Virginia Tech. Dr. Howell received her training at Tulane in neuroscience and cell biology. She went on to Emory for her PhD in neuroscience and completed postdoc fellowships at Emory and University of Minnesota in child development. Dr. Howell's research aims to provide understanding of the complex biodevelopmental pathways through which mothers influence child development and behavioral, especially in high-risk contexts, including low-researched areas, maternal physical and mental health issues. These include variations in maternal behavior and breast milk composition, as well as the microbiome, as well as the effect on the microbiome and its influence on infant development. Please join me in welcoming Dr. Brittany Howell.
Thank you, Dr. Permesar. All right. Well, thank you for that introduction and for inviting me to share some of the work that I've been doing over the past several years and have just started here in my own lab. I do have to disclose that in the past I have been funded by Nestle, but I'm not sharing any of those data here. All right. So I wanted to start, although Dr. P did a great job of giving the summary of what my work does, but I wanted to walk you through it a little bit because I think it really informs how I think about nutrition and brain development. So my lab is really trying to understand this really oversimplified schema of how mom impacts brain development in her baby. And so I break that down into 3 parallel co-occurring pathways. So those include how mom cares for her baby, how she transfers and maintains the microbiome of her baby, and I'm talking exclusively about the gut microbiome here, and then also how she provides nutrition during infancy. So to measure all of these things in the same cohort, what we end up having to do is a lot of different kinds of data acquisition. So we do direct behavioral assessments in the lab of both mom and baby. We do MRI during natural sleep, so no sedation here, just to be very clear. And this is kind of the bread and butter of the lab. That's our primary outcome. We provide a very comfortable and open, welcoming space because what we want is families to have a good experience with us, so hopefully they'll want to come back to see us, as well as to go to other researchers and participate in other research projects. And then finally, I also looked very closely at milk composition, and I highlighted so I highlight the nearest human milk analyzer here because I know I've been talking with some other folks, such as Cameron Johnson, about potentially helping you all using that piece of equipment. So what that equipment can do is tell you the macronutrient and energy density in an individual milk sample. You need 3 milliliters of milk, and then you can see kind of the general quality of that sample. So I just wanted to plant that seed and let you all know that I have the equipment. It's just down the street, and I'm happy to discuss potentially sharing that with you all if that's something that would be beneficial to your patients. All right, so that's how I think about things in general. But I wanted to start today by talking a little bit more about early nutrition, kind of giving things that probably you already know, but again, as a neuroscientist, and you typically are used to talking to other neuroscientists, they don't really think about it unless they have had children recently, and then they know more, they think they know more than anybody else. We're going to talk a little bit about how human milk changes across time. We're going to talk about what brain development looks like across infancy and a little bit past infancy. And then I'm going to make the connection between the two and why I think it's important to consider the two together. We'll go into a little bit about why the microbiome matters and then end with some closing thoughts and takeaways. So early nutrition is often, especially in research, oversimplified to breastfed versus not. And I'm sure you all can appreciate how much of an oversimplification and honestly how hard it can be then to really understand the nuances that surround early nutrition when you do oversimplify and dichotomize that way. So here, what I've shown here is you have breastfeeding at the breast. You can have breast milk or formula out of a bottle. You can have supplemented feeding. You can have, there are different kinds of formulas. There are different feeding schedules. There are all sorts of things, donor milk. And so when we start to think about all those nuances to early nutrition, it starts to become very clear that this is a really complex problem that we really need to understand better. We're just starting to understand this. So hopefully, at the end of this, you all are going to have way more questions about the things that I'm presenting than we'll have answers, which is As a scientist, that's what we do, right? We actually produce more questions than we ever answer, which I know is really dissatisfying as a clinician, but, you know, hopefully you all can take something with that. Okay, so the AAP, their early nutrition guidance is pretty straightforward, and that's for exclusive breastfeeding through the first 6 months of life. That means nothing except human milk until 6 months of age, and then continue breastfeeding for as long as the dyad agrees. And there's huge variation in that how long the dyad desires time. So there are dyads that will stop at 6 months. There are dyads that certainly stop earlier than that. Most do, actually. There are some dyads that will continue until the child is 2, sometimes 3, 4 years old. And of course, you know, everybody has seen, although I think it's kind of the hubbub has died down in the news, there have certainly been cases where women are breastfeeding until a child is 7 or 8, right? But it really doesn't matter. It just depends on the comfort of the dyad. And there's a lot of good reasons for that now. Hopefully, remember to touch on as we continue. This is pretty straightforward advice. And you all, or at least primary care pediatricians, have done a really nice job in the past several years actually supporting breastfeeding by recommending breastfeeding. And so this is a really nice figure that I took, again, just from the AAP. And so between 1995 and 2014, rates of primary care pediatricians recommending breastfeeding within an hour of delivery more than doubled, which is pretty exciting, right? Because we know that that's really important for moms to start as soon as they can and to start building that confidence and just physiologically building supply, right? You're also, pediatricians are also doing a really good job about saying don't give them anything except milk unless medically indicated, right? So sometimes unless you're very explicit about that, that's just not what they're used to doing, right? There are cultural variations in early feeding, and we need to be very clear with patients. Also, increased rates of suggesting rooming in for 24 hours a day, which is right here. I know the text is probably kind of hard to see, but and then unrestricted breastfeeding, so following the infant's cues for when they are hungry, not setting them on a schedule. And then no pacifiers in the hospital. This one is a lot more controversial, so you'll see the rates of that suggestion are much lower. But there is evidence that introducing a pacifier early can reduce breastfeeding success. So. All right, so overall, I want to start positive. You guys are doing a great job recommending. However, do patients always listen to you? I'm one of those patients, so I'll kind of hunker down. But they don't. So don't be overwhelmed by this figure, because it is a little busy, and I'll walk you through it. But I think what's really interesting is it gives you a good snapshot of what early feeding really looks like in the United States. And it doesn't look like what the AAP wants it to look like and what physicians want it to look like. What it looks like is this hot mess. Let me walk you through it. So this dark blue color, this is direct breastfeeding. The green is direct breastfeeding and pump and feed, so this is still only human milk. This purple, or lavender, is direct breastfeeding, formula feeding, and pump and feed. So however I can get milk into or nutrition into my baby, that's what I'm going to do. That's what Baxter is doing. The maroon is formula feeding, and the orange is they don't know because they didn't respond. So at 1 month, you have about a third of babies being exclusively formula fed. That's very counter to what the AAP says and what science says babies need, right? But what's interesting is what happens as those babies get older. And what happens is by 2 months, almost 10% of them have already had some kind of supplemental feeding. So whether that's juice or solid food, something, rice cereal mixed in with their formula, something is being added here, which is much earlier than the 6 months that the AAP suggests, right? So there's something about early formula feeding that then leads to this additional kind of alteration in feeding practice. And so this is SFF, is solid supplementary food feeding. But if you start to look down here at this bottom corner, if they are direct breastfed, then they typically make it to at least, most of them are making it to, at least month 4 before they start introducing solid food. So if we can get them started with direct breastfeeding or even our pump and feed group, then they do much better. I encourage you, I think the recording is going to be made available, and you can check out this, the full paper and talk about, and it talks much more in depth about how they derived all of these things and some of the interesting findings that they had. But the point I want to drive home is that this is not, again, what the AAP wants it to look like. So if this were the AAP, everything would be dark blue until 6 months. And then you would have this light blue, or, maybe dark blue or green, right? So pump and feed, depending on the circumstances, certainly preferable to formula. All right, so despite everyone's best efforts, we still are having a really difficult time getting parents to actually breastfeed. And I don't want to oversimplify and suggest that I think they're just refusing, because that is 100% not the case. There are lots of cultural barriers, there are financial barriers, there are time barriers, all sorts of things. So please don't think I'm saying that patients are terrible and don't know what's best for their children. That's not my point at all. My point is that we need to be doing better to support them. All right, so let's talk a little bit about the specifics of human milk, in case you all weren't already aware of how amazing it is, but if you are, I'll remind you. So human milk is not static, and this surprises most people outside of academia or this area of academia and clinical practice. It's a very, very, very dynamic fluid, so it changes across different time scales, which we'll talk about in more detail in just a second, but the main constituents remain the same. So it contains macronutrients, so things like protein, fat, carbohydrates, so building blocks for babies to grow, micronutrients like vitamins and minerals, which also help with those growth processes. But it also contains bioactives. And what I mean by bioactives are things that actually cause physiological change in the baby without supporting growth. So things like hormones, immunoglobulins, things like that. Milk is really chock-a-block full of those things. And that's one of the things that my lab is really trying to understand is what are those bioactives really trying to signal to the baby, and what are the kind of outcomes related to increased exposure to those things. All right, so as I said, not a static fluid at all. It changes across several time scales, and so the time scale that I'm looking at right now is across a single feed. So in general, you can kind of split milk into 3 different phases during a feed. There's fore milk, mid milk, and hind milk. And this is actually a really old seminal paper, and I didn't put the citation. I can send it to Jamie. But anyway, the point here is that if you look at levels of lactose and glucose, or sugars, they're really high in fore milk. Lipids are low in fore milk. Protein pretty much stays the same, so we're not going to pay much attention to protein. But the interesting part is that as the woman produces milk across the feed, it becomes fattier and less sugary. And so to me, thinking about those bioactives, things that are water-soluble are going to be enriched in fore milk, and things that are fat-soluble are going to be enriched in hind milk. And this also has a lot of implications if you have a woman who's kind of a super producer. So when I worked in Minnesota, we had a woman, so for our research, we collect everything from a single breast with one pumping session. So we asked them to completely express a breast. We had a mom who was able to produce 250 milliliters of milk from a single breast. And so if you can imagine, if she was feeding an infant, that infant is going to get physically full on fore milk before they ever get anywhere near hind milk. And so she had worked with her pediatrician and a lactation consultant to do a timed feed. So she would feed for a few minutes with hind milk, she'd pump a little bit, and then when she noticed the change, because she could feel it actually, which is also an interesting thing, and then she would switch to the baby would get some hind milk as well. But something to think about. So not only does it change during a single feed, but it also changes across all of lactation. And so again, you can split it roughly into 3 categories, but I think we don't know much about milk past about 6 months, because again, it's just difficult to get samples from women because most of them have stopped. But that's job security for me, I guess. But for colostrum, that produced about roughly 1 to 3 days postpartum. So of course, there's variation in all of this for an individual woman. Transitional milk is from the ending of colostrum production all the way to 2 weeks out, and then mature milk is anything beyond that. But again, I think mature milk is probably an oversimplification that we just haven't been able to really dig into yet. But again, if you look at the kind of major macronutrient constituents here, you see some interesting patterns. So again, with glucose, it's really high early, and then right about, let me orient you. So these are days postpartum. So this is about 6 months. This is about a year-ish. So glucose rises, same with lactose, but lactose stays fairly stable across time. Volume increases, that's not super surprising. But what I wanted to point, and protein stays relatively flat after colostrum. So colostrum is packed with immunoglobulins and other kinds of proteins that are really important for training the infant immune system. So that's that spike you see in protein here. But what I wanted to point out are the lipids. And the lipids are the only constituent that tends to increase across time. And I want you to keep that in mind because I'm going to come back to it when we start talking about brain development specifically. All right, so the roles of milk. Traditionally, milk has really been thought to just physically grow babies. And when you think about outcome measures in formula feeding studies or any other kind of feeding studies, that's typically the outcome that's measured, right? Are they growing the way that we would expect them to grow? So what I am going to hopefully convince you of through the next few minutes here is that it's really designed for not only physical growth, but brain growth specifically. And I didn't show any of the comparative work, so looking across species, but if you are interested in milk, you should dive into that because it provides lots and lots of evidence for this very specific role of human milk in human brain development, which is really exciting, at least to me. All of my graduate work was done in monkeys, and so I oftentimes think kind of across species. I think it's important. All right. So let's dive into brain development. And I put all of these pictures here not just because they're adorable and who doesn't want to see cute babies and pregnant ladies, but it's to remind me to say that although early nutrition is a really important aspect of early brain development, it's certainly not the only thing. That's something my lab is really trying to embrace by measuring the million things that we're measuring all at once. So brains are designed, maybe design isn't the right word, but they're really good at adapting to the environment, right? That's the whole point of the brain, is to not be static, to not always do the same thing, but to take in sensory information, figure out what's happening, and to respond appropriately. That's what your brain does. If your brain didn't do that, you wouldn't be very successful. And if you want to, kind of boil it down just to evolution, you wouldn't be able to reproduce very well if you couldn't adapt in different contexts, right? So in order for a baby to do that, they have to learn a lot really, really, really fast, and their brains have to change very, very quickly. So they're doing things like paying attention to mom or other caregivers playing with them. And so face-to-face contact, I have it here twice because it's really, really, really important to actually look at your baby in their face. They've actually shown this in monkeys as well, which is super cool. And then maternal stress, I know this is prenatal, but maternal stress is really one of the strongest predictors of poor neurodevelopmental outcomes postnatally, even with prenatal. So again, just to remind everybody that although nutrition is really important, and I focus a lot on nutrition because it's something that's a little easier to shift, unless you're talking about increasing breastfeeding rates, and then that's not at all an easy shift to make. But we may have some alternatives which I'm going to talk about towards the end. All right. So why do I care about brain development? And I care about brain development because neurodevelopmental disorders are extremely prevalent in the United States and across the world. And we really don't have a great understanding of most of their etiologies, right? We have hints of what risk factors are. We don't necessarily understand how all of these other pathways that I've been talking about feed into these things. And if we could imagine, having a supplement or some sort of evidence-based best practice, we could possibly prevent a lot of these things. And I would argue that a lot of the mental health disorders named here are developmental in nature, at least at some level, right? Again, we haven't done the studies because that takes prospective and longitudinal data collection, which takes a long time and is really expensive. So we just don't have answers to those questions yet. Our funding agencies, including the NIH, are getting much better about funding long-term longitudinal prospective studies, but it's a hard sell when we're going to ask for literally millions of dollars and not come out with an answer for probably 10 years. But sometimes you have to think that far ahead. But anyway, this is one of the many reasons I care. All right. So one of the reasons why thinking early and about brain development and just neurodevelopmental disorders and nutrition is really important is because the brain is most plastic early in life. And so this is a really simple figure that I really love. And if you have patients who show interest in these kinds of things, point them to the Center on the Developing Child at Harvard. So they've done a really nice job of simplifying some of these really complex topics and presenting them in really easy-to-digest ways. So there are lots of simple videos and talks like this that you can talk to your patients with. I use this resource a lot with our research participants. But what this one is showing, and I just, it's so simple and I love it, is that very early in life, because this doesn't start until 2 years, but very early in life, the plasticity or the ability of the brain to change in response to an exposure is really, really, really high for very little effort. And this is great when things are going the way they should, or the way that the infant kind of biologically expects them to. But when things don't happen the way the baby expects, maybe they don't have adequate nutrition, maybe mom, is out, has to work 20 hours a day, and she is stressed out and tired. Maybe they don't have money for food or safe housing. When those things happen early on, the brain is still plastic. It's still going to respond to those things. But what's going to happen is it's not going to develop in the way it would if it were in a more supportive or species-typical kind of environment. And as you get older, the ability for the brain to change decreases. It never goes away, so don't worry. Your brain is always plastic. You can always make it change. It just takes a little more work. That's all. I know the steepness of this is a little scary, but it's also an oversimplification. Point being, though, again, that we need to be targeting things early, and again, that's why I think a lot about very early nutrition. I think it's a nice window to intervene. All right, so I would be remiss if I only focused on postnatal brain development, because it's very easily argued that prenatal brain development is much more traumatic than postnatal. You're starting from a 1-week embryo here and going all the way to a full-term baby here, and their brains look remarkably adult. They're not just little tiny adult brains, just to be clear, but they do look much more similar. But what I wanted to highlight are all the things that are happening prenatally. So you have neuronal proliferation, so the actual neurons are proliferating, then they're differentiating, then they're moving to where they need to be in the kind of the mature infant brain. They're forming connections through synapses. They're then dying if they're not needed, because nobody's got extra energy to support cells that are unnecessary. And then the process of myelination starts. And I'm going to talk about myelination right now. So these are some data that I collected in a single baby between 3 months and 2 years of age. And so I'm sure you can appreciate that this is huge. Just looking at it, even if you don't know what you're looking at, there are massive changes. So the top 2 rows are the traditional kind of structural MRI, so T1 and T2 weighted images. And what I hope you'll notice is the biggest jump happens between 3 and 9 months. And I have full disclosure, we were supposed to get a 6-month scan, but this baby didn't sleep. So we were not able to get good data at 6 months. So you would have seen the big jump then would have been from 6 to 9. But anyway, but what I wanted to talk about when I talk about myelination, and these pictures never show up as well on the big screens as they do on little screens, but is to talk about white matter. So white matter is the connections, the physical connections between cortical regions in the brain. And myelin is the fatty substance that insulates those connections, exactly the same way that electric wires are insulated. But what's interesting about myelin, well, there are lots of things that are interesting about myelin and about white matter in general, is that the cells that produce it, they're called oligodendrocytes. They're precursor cells, or oligodendrocyte precursor cells, don't proliferate and migrate and mature, differentiate until after birth. Some of them start, but most of them do all of that postnatally, which is super cool. And if you remember, I just said myelin is a fatty substance, and I showed that fat actually is one of the only substances to continue to increase postnatally. And so I'm really interested in understanding that connection. What is it about the specific fats that are being produced, or how are they supporting white matter development specifically? All right, so this is just to bring those 2 time periods together to remind us all that there aren't one and then another. And of course, if you're working with premature babies, that's a whole different world, right? So I'm talking about typical brain development here. Preterm infants are another population that I haven't gotten the chance to work with, but I hope to in the future. There are lots of folks trying to dig into what exactly is happening when an infant is born preterm. But just to kind of summarize, so you have all of these neuronal processes happening prenatally, and then postnatally you have all of this myelination, and of course synaptogenesis continues to happen. I do want to mention that myelination is not static. So just like breast milk was once considered just to be one kind of thing that just grows babies and that's it, you're done, myelination was thought to be a process that you did and then it was over. That's 100% not true. It is a dynamic process that's driven by brain function. So if you have cortical regions that are more active together, and those physical connections, the white matter that connects them, will become more myelinated. They've shown that in some really interesting ways, like teaching people how to juggle, which is kind of fun. And their white matter tracks that support coordinated hand movement actually increase myelin. And that's an adult, so you can learn how to juggle too, guys. All right. So what is the milk-brain connection specifically? And there are lots. There are lots and lots and lots. Let's go over just a few. So one of the kind of grossest measures that you can look at is brain weight. This is just gross brain weight across time. So first, let's just look at these plots, or this plot rather. So at birth, you're right here, about close to 400 grams, and then it increases until about 2 when you're a little over 1,000, and this, for reference, is just what the average adult brain might weigh. The point here is if you look, so each of these are a little confusing. So each of these breaks corresponds to the weight of the brain at that time. So let me walk you through. So this bar is a baby that was exclusively breastfed for 6 months. So if you look at that, that baby's brain is going to be about here, weight-wise. So that means that a lot of weight, which is represented by the height of this bar, a lot of brain weight was gained while that baby was exclusively breastfeeding. The amount of weight in the brain weight that the baby gained if they were only breastfed for 4 months is drastically different. And so what this is suggesting, or what this is showing, is that the longer that first 6 months is actually really important just for brain growth and exposure to whatever is in milk that's supporting brain development. So kind of a complicated figure, but again, the idea here is that you actually, if you're breastfeeding for a full 6 months, most of that baby's brain growth is going to have occurred over that.
How's that data obtained? Is that extrapolated from MRI, and do you have just 2 patients represented there, or you have a cohort?
So this is average. This is kind of a theoretical plot. I don't think she used any actual weight. I think she did kind of a meta-analysis of kind of average typical weight, just to make the point that lots of brain growth, just growth happens during those first 6 months. But so that's a good question. And this is an older paper. This is older data that she's using. We typically don't even care about brain weight. We typically, at least for an MRI, we just stick to what we know we can measure. So we'll do things like cortical thickness and surface area and volume. All right, so there is correlational evidence in humans, because a lot of the time we can't do, controlled feeding studies are hard enough in adults, never mind in babies, unless you're doing a formula trial, and then it just takes a lot of money, and lots of formula companies have access to those kinds of resources. But if you just do a really simple analysis, looking at how white matter, and this is volume of white matter, or how that is related, to breastfeeding duration. There's a really strong, and I don't know if any of you have done research, but an R squared of 0.32 in any kind of imaging study is massive. So this is a really cool study that looks to see whether or not I believe these kids were 2, whether or not their white matter was different and whether that was predicted by the length of breastfeeding. And it absolutely was. So again, this is suggesting that white matter specifically is really influenced by at least duration of breastfeeding. So this is how a lot of the studies are being done. They just will retrospectively ask, how long did you nurse your baby? What my lab is really working hard to do is get at a really granular, how did you feed your baby? So were you feeding at the breast? Did you have to pump and feed? How did you feel about your breastfeeding experience? Did you really like it, or did you just do it because you thought it was what was best? But we're doing that because there is this, even if you look, just kind of scratch the surface, there's a pretty strong connection here. All right, but I'm very doubtful that any of you have had a patient come in and say, I'm pretty sure my child has too little white matter, or myelin. Maybe someone has come in. But anyway, they're probably going to come in and say, you know, there's a behavioral issue. And so we also, as researchers, and this is some work that I recently published with collaborators at UNC and Minnesota, we want to understand the endpoints. What kind of behaviors are actually related to things like breast milk and the things that are in breast milk? So what we found was if you look at, this is a receptive language score from the Mullen Scales of Early Learning, that is positively predicted by concentrations of DHA, so everyone's favorite formula additive, DHA. This is phosphatidylinositol, and then total short-chain fatty acids were the best predictor. And keep that in mind as well, because I'm going to bring that up again in a second. But my point is that these are all fats that are feeding into our receptive language score. So there's a behavioral consequence, it seems, to having different compositions in milk. I do have to point out that this is a typically developing population, and the variation that we saw was a typical amount of variation. So none of these kids were at a deficit, and I don't believe we had any that hit ceiling. So take that, for what you will. But these are very happy, healthy, typically developing children. The bottom is just showing this some aspect of temperament, so surgency, so kind of think of that as baby extroversion, is also predicted by milk components, but these are arachidonic acid, phosphatidylinositol, and then phosphatidylcholine, so micronutrients, and then another fat. But anyway, milk matters for behavior, which is really exciting to me, but now we have to figure out why and how and what is driving concentrations of those things in milk. That is a totally open question. We don't know if is maternal health driving it, is context driving it, and honestly, it's probably going to be a mixture of all of those things, and we're not there yet. We're working on it. All right, so again, I said milk helps grow babies and grow babies' brains, but I wanted to introduce this idea as milk is a signaling fluid. So what milk can do, it's in a really unique place to kind of filter what mom's body thinks is important and give that information directly to baby. So one of the ways that it can do that is through hormones. So this is a single example of one of my favorite hormones, because all of my past work was done in early life stress and stress physiology, so I can't do a talk without talking about cortisol at some point. So cortisol is the end product of the hypothalamic pituitary adrenal axis. It also plays a very large role in just regular energy metabolism throughout the day, which is why it has this beautiful diurnal rhythm. So these bottom bars are milk cortisol measures, and these open dots are mom's salivary port. And so what they were able to show here is that there's a really good correlation between milk port and mom's salivary port. And they have not done this study under stress, so it's still kind of an open question about whether or not if mom is stressed, so her port levels are up, if that's reflected in milk, because there's lots of evidence that excess exposure to port in baby is actually not great. And it would be very interesting to see if mom's body actually, kind of dampens that and acts as a buffer so that baby is not exposed to those increased levels. We don't, again, totally open question. Also confounded that when you're stressed, you don't produce as much milk, right? Breastfeeding is not as smooth a process. So we're working on it, again. It's a process. But what I wanted to point out, because I'm super excited about this, are milk exosomes. So I don't know if you have dived into the cancer literature, then you've probably read a little bit about exosomes. So they're these really tiny, 400, 500 nanometer diameter particles that are used for intracellular signaling. So tumor cells do this. Most of the cells in your body produce exosomes. Mammary epithelium cells, so the cells that actually produce milk, are great at producing exosomes. And up to 20% of the volume in human milk is exosomal. And of course, that varies across lactation and other characteristics that we're still digging into. But mom is putting a lot of energy into producing exosomes. And what's interesting about exosomes is that they are not packed via diffusion like other vesicular structures. They actually seem to be packed through an active process, which is, again, super cool, and to me, puts them in a really great place to be the messengers that mom is sending, one of them, many, that mom is sending to baby. They typically contain RNAs, primarily microRNAs, and then peptides. We're just starting to understand this in human milk. People understand this fairly well in bovine milk, because the dairy industry is actually massive, and there's a lot of evidence. We just looked at a paper in lab meeting that said that milk exosomes in bovine milk are actually what's leading to all Western disease. So if you want to be scared about drinking your milk, you should read that paper. It was not sponsored by any dairy council. But what's interesting about, well, one of the many things that I find extra interesting about exosomes is that they actually accumulate in the brain. So they've shown this in animal studies. We haven't done any, because we can't do anything in humans as of yet. There are no safe imaging techniques, but we know that they accumulate in brains. And so this, again, puts them in a really good place. They contain signaling molecules. They survive the gut. They make it to the brain. But what, again, is it? What's driving what's being packed inside them, and what are the consequences? And we don't know. So I'm putting in a grant, hopefully, so this is actually data that came out of milk from our lab, and we have a small army trying to optimize some protocols so that we can apply for some funds to look at what is driving exosomal content. Because we don't know. We know that they're there, we know they have this stuff in them, but nobody's looked to see what is it about mom or the environment that's, kind of writing the message, so to speak. All right. So I'm going to jump into the role of the microbiome, because again, I can't not talk about the microbiome and talk about nutrition. And this has been super hot recently. I'm sure you guys probably get questions all the time about, should I give my baby probiotics or prebiotics? All of those kinds of good questions, right? All right, so when I talk about the microbiome, I'm just talking about the gut, but the microbiome is certainly complex, so you have a different microbiome on every different part of your body. It includes bacteria, which is what I'm going to talk about, but it also has fungi and viruses, archaea, all of those things are somewhere on you. And I'm sure you've heard the really kind of scary number that you're only 10% human, and that's just by really rough cell count, so don't be that scared. You are yourself, don't worry. They're really important for food digestion and immune system function and immune system development, especially in babies. And they really need to be kept in balance to maintain health. And what I mean by balance is there are several different general classes of microbes. So there are symbionts, so those are the things that actually benefit you. There are commensals, which, take them or leave them, they don't typically make much difference. And then there are the pathobionts, right? So those are the things that can cause disease. So normally when these things are in balance, you get, these are regulated so that you have just the right amount, and they actually reduce the ability of the pathobionts to colonize, right? As soon as those symbionts get too low, that allows pathogens, these bad bugs, to kind of take over, and pathogens can come in, leading to lots of inflammation. That's one single pathway. There are lots of others. This is, again, oversimplified. But the point is that we really need to have balance. What balance looks like seems to be very individual. It depends on age. And there's still, that is a very open question in all of science right now, never mind just for babies and children, but for adults as well. Lots of interest for obesity specifically in this area. And why do we care? Well, it's because dysbiosis in the microbiome has been found in lots and lots of disease states. So there are the obvious ones like functional GI disorders, there are metabolic disorders, right? Those kind of make sense. But then there's also neurodevelopmental disorders, and addiction, and stress, and neurodegenerative disorders. One of the big questions that's kind of out there, though, is whether or how diet interacts with these dysbioses, because it's like autism is a perfect example, right? There's lots of work coming out that's suggesting that alterations in the microbiome are actually part of the etiology of autism. The evidence is not really strong if you dig into it, because no one has done a good job of factoring in the fact that a lot of those kids are only eating, 4 or 5 kinds of food, and that's definitely going to impact your microbiome, right? And those are things that we can factor in statistically. It just takes huge samples. So hopefully somebody at some point will look at those things. But anyway, this is why we care about the microbiome. Why I care. I care especially because there's lots of actually very strong and compelling evidence that that establishment of the microbiome is really important for infant development. And so this is just, again, an oversimplification of these complex things. But we know that mode of delivery impacts, so whether a baby was vaginally delivered versus cesarean delivered, and that early feeding practice, so all the things that I was just talking about. And so this is just a kind of a pictorial view of what a vaginal-born breastfed baby's microbiome would look like. So notice that there aren't a lot of different kinds of bugs, and there aren't a lot of them early on. Once they hit about 4 months, and this is when they're really good at getting those hands in their mouths and crawling around, and they're probably, they may be in daycare, so they're being exposed to a lot more things. So their microbiomes become more abundant as well as more diverse, and that continues until they're about 12 months. A vaginally born bottle-fed baby actually has a more diverse microbiome earlier, and there's some suggestion that this increase in diversity is actually not good for the baby, and there's been some links with decreased diversity and better cognitive outcomes later in life. This is one of the few places where we actually don't want more diverse. I'm sure if you've listened to any news or radio, it would eat a lot of different kinds of food to make your microbiome diverse. That's true in adults, it's not true in babies. And then if you have a C-section baby, they're colonized by completely different bacteria because they're not exposed to the vaginal canal and on all of that wonder, the wonder that is childbirth, right? And then by the time, though, that they get to be about 12 months, diversity-wise, they're not distinguishable, but they're certainly still different kinds of bugs. Again, this is, when you get these data sets back, you get thousands of different species of bacteria. And those, again, they vary by season, they vary by part of the world, and so we're still, as scientists, trying to figure out what it is specifically about specific kinds of bugs and how we can best support families in making good choices. Because we know there are differences. We don't necessarily know what the consequences of those differences are, and we don't necessarily know how to shift them if we need to. But milk is potentially one way that we can guide the infant microbiome. So on the panel on the left here, this just shows, that there are lots of changes to the breast in preparation for lactation, including potentially increased permeability. And that's important because there's been evidence that in the breast, in the mammary epithelium, that the dendritic cells of the maternal immune system will actually actively pluck certain bacteria from the maternal gut lumen and shuttle them actively to the mammary epithelium. So mom's body is kind of curating the kinds of bacteria that baby is exposed to and breeding them in her milk. And there's also this really amazing bidirectional interaction between the breast and breast milk microbiome and the infant's own oral microbiome. These are all just starting fields. We have no idea kind of how to guide anybody towards anything, but we're looking. And what I wanted to point out, and this is the point that I always make when I'm asked by participants whether they should be giving their babies prebiotics or probiotics, is that if you want a bacteria or a fungi to survive in you, you have to be feeding them, right? You have to be giving them food that they can digest. And for the kinds of bugs that we see in babies' guts that seem to be particularly important for development, so things like Bifidobacterium infantis, they do really well digesting human milk oligosaccharides, which is shown here in the palest wedge, this pie. And you'll note that it's the third most abundant component in breast milk. So these are specific carbohydrates that baby cannot digest directly. They're indigestible by baby. However, gut bacteria, like Bifido, are great at digesting HMOs. So my point is when somebody asks me, should I be giving my baby probiotics, my answer is, well, are you breastfeeding? Because you can't get HMOs anywhere else. They're human, so they're only in human milk, and they are not easy to produce to scale, which is why most infant formulas do not have them. There are some that are spiked with a little bit, but not nearly the same levels that you would see in actual breast milk. But that's honestly not that surprising to me, where most formulas are not even lactose-based, even though human milk is. So we have a long way to go with that as well. All right, so I have to show you some of the work that my lab is doing, and this looks like a really boring figure to most people, but I was excited about it. So this is a really deep dive into the bacteria in what we're calling the breastfeeding microbiome. So in my lab, we don't sterilize the breast or the nipple because what I want to see is what baby is exposed to when they're nursing or if mom pumps what she's providing for baby. And so what we actually find is that, and this isn't super surprising, but we still don't really understand why, they're primarily exposed to skin bacteria. Okay, that doesn't, why? That doesn't make a ton of sense. There is certainly, so each of these is a single participant, there are 15. And we have samples here from 1 month all the way to 10 months in some of them. So there's lots of variation. We did not find any patterns across lactation. There were not patterns within each woman, at least for bacteria. And then this is just, again, to appreciate the variation amongst women. So I know we're trying to see, are there patterns across time? And there may be if we're able to add some of those other determining factors. So, you know, seasonality is going to be a huge issue for these kinds of samples. And again, that folks haven't had the resources or just the sheer numbers to get at some of these questions, but there's lots of variability here. All right, so to sum it up, there are lots of ways that the gut microbiota can impact the baby's brain, and these same mechanisms do exist in adults as well. But they can themselves produce neurotransmitters and hormones. Babies' guts are leakier than adult guts, right? So they're potentially more, it's more easy for the factors that the microbiota are producing to make it to the brain. And I planted the seed earlier about short-chain fatty acids, if you remember. And a lot of the bugs that are important for the baby's immune development and what we're proposing is brain development as well, are actually great at producing short-chain fatty acids, especially when they're given human milk oligosaccharides. So hopefully we're making some connections. You get human milk, you have a good microbiome, they produce lots of short-chain fatty acids, which can make it to the brain and support healthy, at least white matter development. Other membranes can also be impacted by those things as well. And then there's also a direct connection to the vagus as well, in the enteric nervous system. All right, so to sum it up, what I care about, what I'm calling the gut-brain axis, and some people will call this the gut microbiome brain axis, because you don't want to downplay the importance of the enteric nervous system itself. But is that all of the things that I talked about happening in brain development are happening at the same time as development of the microbiome. Now, whether or not this is just a coincidence, or whether or not this bidirectional communication is important across time, or maybe particularly important at certain times, we don't know yet. Again, because this takes longitudinal prospective data collection, and that, again, is really hard to get funding for. So I don't know if any of you have the ears of any policymakers or anyone at the NIH, but please put a plug in for us, because we're trying to answer some really important questions. It's just it takes a lot of resources. All right, so at the end of the day, what we're really trying to do is take a dysbiotic microbiome and add something to it based on the information that we can get. So looking at dysbiosis triggers and then what we identify as a healthy microbiome, so that we can hopefully reestablish a healthy microbiome. The piece that's not shown here is timing, right? So is it more important to do this earlier? I always argue earlier is better. We have no evidence that that's not true in any case. In every intervention that I've seen, the earlier you can catch a child, the better. But you know, maybe I don't want to make any assumptions. All right, so closing thoughts. So is this the future of infant nutrition? So there are a couple biotech companies, one in North Carolina and one in Singapore, that are producing human milk in a lab. And so what they're doing is they're taking bioreactors that are typically used for producing things like antibodies, that's what's pictured here, and they're culturing mass quantities of mammary epithelium cells and then inducing them to produce milk. Or as Biomedical likes to say, milk with a Q. I kind of like that because it's not, they recognize that it's not exactly real human milk. But this is just starting. So macronutrient-wise, it looks very similar. Looking at things like signaling molecules or those exosomes, no idea. We're still digging into, well, I'm not part of this, but they're still digging into it. The questions, again, that I brought up about what drives the kind of composition of milk it's going to be really important for something like this, because you can raise those cells in whatever environment you choose, and that could potentially impact the kinds of milk or the quality of milk that they're producing. But imagine if you could make mass quantities of colostrum for NICU babies. Imagine if you could make, really, if there is a, baby that's past, that's the 2-week mark, but is feeling, mom is sick, baby's sick, and you can give them milk that has an increase in immunoglobulin, right? Imagine immunizing children using milk that was produced by cells that can produce specific kinds of antibodies, right? So there's a lot of room here, I think, for engineering, but we don't know exactly how to engineer that just yet, but people are working on it. All right, so summary of key points here. Again, I hope I convinced you that milk is really important, not just for physical growth, but for neurodevelopment as well. Milk is dynamic, and it changes composition to meet the needs of the growing baby. There are lots of ways that milk can support brain development. I just showed a handful. There are a lot more questions and answers at this point, but we're really doing our best to answer them in a way that is impactful and informative for you all treating patients and for families with typically healthy kids. And then, again, my lab is working really, really hard to get the strong and robust empirical data we need to inform best practices and therapeutic efforts. So I can't leave without thanking everyone in the lab, and this isn't the most updated picture. We need to get another picture. But anyway, the lab has grown almost exponentially. I've been here for about 2 years, and we have lots and lots of really eager and skilled hands helping. And with that, I'll take any questions, but I have to point out, this is my daughter, not this one. This is my younger daughter at 10 weeks old. So of course, she had to be in a study, right, because that's what we do. And then this is my older daughter when she was about 4, and she's standing in front, we went to the Science Museum, and she's standing in front of a connection shaped the brain, so something talking about actually white matter. And she happens to be wearing her T-shirt that says mostly microbe on it, so I can't not show that. Aww. I know, right? Okay. I'm happy to field any questions you may have.
Yeah, because I find it very interesting about trying to actually manufacture human milk when each mother's human milk is so unique for that baby.
Absolutely, yeah, you're exactly right. And that's going to be a big challenge. I think where they're coming from, though, is that they could potentially produce something that's more similar to human milk than current infant formulas, which are not at all. But the vast majority of them don't do a great job at all of mimicking.
Yeah, because I can't get excited about 2FL and the HMO added to one of the infant formulas because not every mom makes it. And and there are, what is it, 160 some probably?
So 200, right around 200 now is what they've identified. Well, it's interesting, so 2'FL is interesting because when a woman does produce it, it's the primary HMO typically.
Yeah.
That's by volume, that's the one they're producing most, but you're exactly right. There are non-secretors, right, that don't produce it. And so people have looked at that, and there doesn't seem to be much impact on the baby specifically. Like you can't tell a non-secretor's baby from a secretor's baby later. So yeah, we're not sure, but I agree. I wasn't that excited when I saw that.
That's good, because I say when you make a living dynamic fluid, then come talk to me.
They're working on it. They are. Somebody showed something else.
Are you having
I'm sorry.
Are you having any data yet to show whether there should be like a longer time of breastfeeding than 6 months that the current guidelines say?
That's a great question. So the question is, do we have any evidence that kind of supports that 6-month cutoff? And the answer is no. And the reason that I think the answer is no is because the outcomes aren't clear, right? So is it just physical growth? That's the most important outcome. Is it brain growth? Is that the outcome that we're interested in? Is it behavioral outcome? Right? So those things I think are really, we need to answer those questions first, and then I can design a study. But I can't design a study to tell you when to stop breastfeeding if I don't know what the what optimal outcome looks like. The way I always talk about it, and I'd love to hear from you all about how you talk about it with your patients, because I'm not a medical doctor, but we get asked questions all the time about how long should I breastfeed, is it better to feed at the breast versus with a bottle? And my response is, first, talk to your physician. I do say that. And then the second is, do what you feel is best. And I think, so from the research side of things, if you have a confident parent, then you have somebody who is at least, number one, willing to listen to you when you are presenting them with evidence, right? And they are willing to try something different and potentially continue. And so that's also something, so when I'm talking about what's kind of determining what's, what's being produced in milk, this idea of maternal agency and maternal stress is really important to me. And so we're working hard to really try to narrow that down. If you look at breastfeeding interventions and things like that, they're most successful when mom gets, even if it's just her body's ability to start to produce milk. That bolsters her confidence and her agency and her self-efficacy. And so if we can boost those things, independent of things like breastfeeding, then I'm guessing that we'll have better, it will have better breastfeeding rates and we'll have better infant outcomes. But no, that is a great question. So, and I wish I had an answer to it.
So this might be kind of a weirder question, not as straightforward, but this is something I've been pretty curious about for moms who have multiple children in different ages. So if you have a mom that's breastfeeding kind of maybe like a little bit over a 1-year-old and then has a newborn, and breast milk being a dynamic fluid, how do you think that?
No, this is a great question, and scientifically, I don't know what people have looked at. Anecdotally, and talking to women and looking at what's been published, the milk will shift to support the younger child.
Okay.
And so we've had lots of families in studies, so we typically will get families that are on either their first or second child, and by the time, so we're following that's the one that's enrolled until they're about 2. And so then typically moms, if they're going to continue to grow their families, they'll get pregnant again.
Right.
And there's always this struggle towards the end of the pregnancy when colostrum starts to come in,
right,
because it's thick, it's sticky, it's sweet, and the older child loves it. And so then as soon as the baby is born, it makes that transition way more complicated and harder. Yeah, so it seems like the milk is dynamic and it does adapt to the younger child. Yep.
Okay, thank you.
Sure. Dr. Keaton.
This is fascinating, and thank you so much.
Oh, sure.
It sounds, most of your talk was really focused on milk composition, but I don't know how you tease out the interaction between the behavioral part of breastfeeding, because you're getting contact, skin to skin with the mom 8 to 10 times a day, often facial, the behavioral part of it. So that clearly stimulates the child's brain to grow. So the multivariate analysis that you have to do here blows me away. And any comments on that?
No, so I'm in that same boat, and this is why I'm really pushing for funding of large-scale studies, because it is important to know what is the impact of feeding behavior. And so we actually in the lab, at least pre-COVID, when we were going to have families come in, we were going to record a feeding session and actually code it to look at things like face-to-face contact, how often were they skin to skin, all of that kind of stuff. Because you're exactly right, and there's lots of so animal work, the way that they don't exactly mimic it, but they'll use a paintbrush to tactilely stimulate babies or pups to try to, again, in a controlled way, mimic that kind of contact. And you're exactly right, it 100% does. There's also all sorts of confounds when you talk about mom or dad working, right? So, it's very different to be handled and fed by a child care worker, right? Not that they aren't amazing. Don't get me wrong, I love daycare workers. They are amazing people. But they're not mom or dad, and we know the brain distinguishes that and changes differently in response to contact from mom to dad. So, there are even these bigger layers, which is exactly what we're trying to get at. My lab itself can't do it alone, and so we have lots of collaborators across the country, and we're trying to expand to work with some folks in Finland, because Finland, as everyone knows, is the happiest place on earth, right? If you want to have a baby, you should have one in Finland, and they're doing lots of similar work. But no, you're exactly right. You are 100% right. And we're trying to get that evidence so that we can push for longer maternity leave. That's my side hustle, is trying to show pretty pictures here to be like, you should give us a full year to take care of our babies. But we're slowly, but I don't know, surely. Just the slow part.
That's right, looking at the difference between feeding breast milk in a bottle versus directly fed.
Absolutely, and we actually have some experiments coming up where we're going to look at brain function during those different things. All right, and then I have to ask your board-relevant questions. All right, shout it out, guys. So what is the compositional forms of breast milk as currently defined are? Good job, yes, B, very good. All right, And then exosomes in milk are A, up to, oh, keep reading. I won't read them out loud to you, you already know.
D.
Very good. And I think amazing should also be on there. They really are. All right, and I see one question in the chat. Just the one? Oh, no, we have two. Oh, goodness, okay. Okay. So very interesting. Strong recommendation. Breast milk preterm infants do have good evidence to support that. They absolutely do. But our studies done in the past beyond observational studies on breast milk that strongly support breastfeeding, RCT. Oh, that's like, so not that I know of, because you can't randomly assign kids to breastfeeding or formula, because we know that breast is best, right? So there's an ethical concern there. I'm trying to think if there have been any animal studies, and it's hard because then you have the huge confound of having them away from their mothers, right? You can't, at least I'm not aware of any animals that have been trained to feed their babies formula. We can talk about it. I'm up for weird ideas, but no, so I don't think there have been any RCTs. Any mothers who have had negative experience with the approach of education for breast past beyond observation. Oh, wait, hang on. Sorry. I have also seen mothers who have had negative experience with the approach of education for breastfeeding by healthcare teams underevaluated. Yes, it is an underevaluated issue. Absolutely. So I'm working with Dr. P and Dr. Johnson to hopefully put together a more grassroots approach to breastfeeding support and focusing on community engagement rather than us as the experts telling them what to do. That's one of the things we're trying to do. Oh, and how do we know breast is best? Google it. Awesome. All right. I think I'm being told to stop.